Hello again. Today I want to talk about cell division and how it's regulated, and then we'll talk a little bit about how that affects cancer when the cell division is misregulated. And when we talk about cell division, um, multicellular organisms are quite a bit different than single-celled organisms. With a unicellular uh, organism, it basically grows and divides when it's happy, when there's plenty of nutrients and energy and in favorable conditions, and then slows down when those conditions become less favorable. Um, and we talked about this a little bit, that there's a natural selection for those that divide more. And so if you have a population of, say, bacteria in a culture, and you have a mutation in one of those that allows it to grow a little bit faster, it will very quickly overtake the rest of the population. And the faster it grows, the more divisions, the more mutations, so the faster things are going to change. Now, when single cell no longer divides, that's when we consider the organism dead. So really, the viability in this case depends on their ability to grow and divide. Now, in multicellular organisms, that's quite a bit different. In a mature organism, the vast majority of our cells are not dividing, regardless of the nutrients or other conditions. And some cells are able to start dividing under certain conditions. Other cells are terminally in a resting state. And there are a lot of different controls that control different types of cells. So there are many signals going on that tell certain types of cells to divide, while other has no effect on most of the cells in a complex organism. When cells can no longer divide, then they are generally replaced. And this is even controlled very uh, uh, controlled manner that we'll talk about in a few minutes. Um, let's look at what's called the cell cycle. Um, and so, again, if cells are dividing, they're going through this cycle. And right away, we see off to the right here what's called G0. And that's where resting cells are. So if cells are not going through the cell cycle, they're in this state called G0, where they're not growing and multiplying. If they're going through the cycle, we can start in G1. And this is a period where the cell has probably just divided, and it's getting everything together, so catching up on all the things it needs to be a functioning cell, and getting ready to move into another round of replication. And once it's ready, then it goes into the S phase, uh, which stands for synthesis, meaning DNA synthesis. So this is a period where the genome of the cell is replicated. Once it finishes that process, it goes into another G2 phase, and G's in both these cases originally stand for gap, because these are named by people very much involved in DNA replication. So since things didn't seem to be happening at these times, they were called gap 1 and gap 2. But during G2 is when the cell gets ready to actually divide, and then it goes into M phase, or mitosis, where the cells actually line up, condense the chromosomes, line them up on a spindle, and assort them non-randomly so that each daughter cell gets a full set of the chromosomes to make the entire genome. So even though this circle kind of shows each phase of the cell cycle as being about equal, that's certainly not the case. Mitosis is much shorter than the other phases. G1 can vary quite a bit from a short time to a very long time, as well as G2. So depending on the cell and the conditions, those the gap phases may vary quite a bit, but S and M are generally pretty similar in every round of the cell cycle. Again, where M is very short, S phase can be quite a bit longer than that. Now this is a very simple diagram of the cell cycle, and we'll be looking at it in more detail in a few minutes when we talk about regulating the process. So a lot of what we know about the cell cycle was learned by studying cancer, and we have a group of genes that are involved in regulating the cell cycle, and those are normally, in their normal state, are called proto-oncogenes. So these are genes that can become oncogenes, in other words, can become genes that promote cancer, but in their normal state, they function to control the cell cycle or cell division. So proto-oncogenes are dominant from genes. In other words, if we have conversion of one proto-oncogene to an oncogene, that can promote cancer. And again, the oncogenes are a mutated proto-oncogene that now is basically telling the cell to divide more often or than it should, or when it should, or to divide when it shouldn't. Now there's another set of genes called tumor suppressor genes, and these are anti-proliferation genes. So these are telling the cells not to divide, and these that would be recessive. You have to have two tumor suppressor genes malfunctioning in order to get the response. So if you have just one good tumor suppressor gene, it will function to control cellular proliferation. So you have these two groups of genes, one telling the cell to divide and one telling the cell not to divide. So when the cell is going to grow and divide, it needs to turn off the suppressor genes and turn on the signals to tell it to divide. If it should not be dividing, you'll get the opposite happening. The suppressor genes will be active, telling the cell not to start the process, and the proto-oncogenes will not be able to start the cell division process. Okay, if we look at the cell cycle, a little more detail here. Again, the G standing for gap, G1 right after the cell divides, and G2 between DNA synthesis and the actual division or mitosis. Here we divided mitosis, the division step into mitosis, which is the again the condensation of the chromosomes and lining them up and moving each set of them into opposite directions, and then cytokinesis, where the cells actually split apart, or the parent cell actually splits apart into two daughter cells. And here a little bit of timing. So the cell cycle can be anywhere from 12 to 24 hours, and again, you're going to find many exceptions to that, whereas mitosis takes much shorter period of time, about an hour at the most. During early development in insects like Drosophila, and actually cells haven't formed yet, so there are just nuclei, and those nuclei can divide about every 5 minutes. So that's really an efficient working of the cell cycle. But certainly after very early in development, that slows down to a more normal pace. Now again, the vast majority of cells in in our bodies in a complex mature organism are in the G0 phase, so they're not actively dividing. And like I said, some of those cells are terminally in that phase, can't go back and start dividing. Other cells are able to re-enter the cell cycle under the appropriate conditions. And of course, we don't want all our cells to continue to divide. That would create a huge mess. And again, in G1, this is when the cell has just divided, and now it's going to duplicate its contents, so it becomes a normal functioning cell. So this would happen if the cell was going to go into G0 or not. Now in S phase and then going into G2, the chromosomes are duplicated and checked for errors. Everything else is checked to make sure got everything and ready to move into mitosis, where the cells actually divide. Now there are a number of checkpoints in the cell cycle, and some of those are shown here. As you might imagine, down in the bottom right-hand corner, there's a G1 checkpoint. So this is, again, telling the cell really whether it's going to go into S phase or whether it's going to go into G0 and stay in cell cycle arrest. There's another checkpoint in the G2 to make sure that everything is okay and everything's replicated, so that it's okay to go on into mitosis. There's also another checkpoint to make sure that everything's ready, and the chromosomes are attached to the spindle before they are separated into two daughter cells. Now during this process, there are a number of signals. The cell is constantly monitoring to make sure that RNA and protein is enough for the cell to carry out its normal functions. But there are also a number of signaling molecules, hormones, cytokines, growth factors in particular. And again, these are all really proto-oncogenes that can be mutated into an oncogene under certain conditions. So a lot of these checkpoints are controlled by what's often abbreviated PTKs for protein tyrosine kinases. And these are signaling molecules. They're enzymes that phosphorylate other proteins. So these are enzymes that transfer the gamma phosphate from an ATP, that's the distal phosphate, to the tyrosine on a target protein, and that thus activating, in most cases, or in some cases inactivating that protein. Now, these targets could be intrinsic. In other words, it can phosphorylate itself and activate a site on itself that will stimulate uh, uh, the process, or it can be extrinsic and phosphorylate another protein, and we'll show you some of that in a minute. So this diagram depicts the reaction of the tyrosine kinase, and on the left you see the protein with a tyrosine on it, and then this is phosphorylated using ATP and transferring the gamma phosphate to that tyrosine to generate ADP and a phosphorylated substrate. And again, this can be another protein, or it can be phosphorylating itself. And so in this example, we see, and this is the case, generally the case, that this process starts at the cell surface where a signal molecule comes in and binds to a receptor, and that usually causes aggregation of the receptors and activates the kinase activity, in this case phosphorylating itself at different tyrosine residues. And once that happens, then that the signaling part of the molecule becomes active and signals downstream proteins to start the cell division process. So this is a list of different tyrosine kinases, and you can see there's a long list of them, and certainly is not exhaustive. We're not going to go through these or memorize them or anything, but just note that as you can see in the right-hand column, that different kinases affect different tissues. So there are a large number of these that affect the proliferation in many different types of tissues. So we have a set of growth factors now. These are signals for proliferation that bind to receptors on the cell surface. Usually it takes several different factors to initiate cell division, and that's a good thing, because if it only took one, then a single mutation could lead to a cancerous situation. Some of these only induce cell growth but not division, but they'll be combined with others that will induce division as well. And again, these bind to specific cell receptors and initiate a signal transduction pathway, which we'll see in a minute. And this one example that's a nice paradigm for growth factors is the platelet-derived growth factor, or PDGF. Keep in mind that a lot of these things are named from where they were due to where they were originally found, but that doesn't mean they're unique to that particular tissue. In some cases they may be, in other cases maybe not. And we'll see a good example of that later on in a gene that's called the retinoblastoma gene, which is found in pretty much all tissues, not just retinoblastomas. So when the PDGF attaches to the receptor on the cell surface, the first thing that happens is increases intracellular calcium concentrations. You get a reorganization of actin filaments that facilitates attachment, and then activation and nuclear translocation of transcription factors. So these are transcription factors that are sitting out in the cytoplasm inactive, and when they get the signal, they move into the nucleus and initiate transcription, which leads to DNA synthesis and cell division. So this is the process here shown. We have a growth factor, PDGF as an example, binds to a cell surface receptor. You get aggregation of those receptors, and then that activates this kinase activity, in some cases intrinsically, other cases extrinsically. So then the phosphorylation at the tyrosines results in interaction with other proteins that then recruit these signaling enzymes that ultimately lead to activation of the genes responsible for initiating DNA synthesis and ultimately cell division. So this shows a schematic of growth factor receptor, the receptor part of the molecule on the outside of the membrane, and then the kinase domains on the inside that then recruit some of these other downstream proteins shown on the right, like SARC, P85. The P85, when you see this type of nomenclature, generally means the P means it's a phosphorylated protein, which is what we'd expect here. 85 is likely the estimated molecular weight of the protein at 85,000. So these are a number of other downstream factors shown here. This is a diagram of the function of PDGF, again binding to the receptor. In this case, transferring the signal to kinase, to the kinase RAF here through a RAS protein. And then that results in this cascade of kinases resulting in activation of MAP kinase, which then can go in the nucleus and activate transcription. I think Dr. Bell talked about MAP kinases, so I'm not going to go into much more detail here. These RAS proteins are GTP binding proteins that, again, transfer that signal from the receptor to a tyrosine kinase, shown here on the right. It's active when it's got GTP and inactive in its GDP form. But any one of these molecules throughout this pathway can be converted from a proto-oncogene to an oncogene in a number of different ways. For instance, if you have a RAS protein that is always active in signaling RAF to start the process, then that can be oncogenic. Or if you have a receptor that is always active in always signaling the RAS to send the signal on to RAF, that again is oncogenic. And in fact, if you increase the number of receptors on the cell surface, then you can, the cell will respond to a lower concentration of the growth factor than it normally takes to start the process. So many ways to convert the cell from a proto-oncogenic state to an oncogenic state. And this is shown here in this diagram where we have a cytokine binding to its receptor and stimulating JAK, which recruits the STAT, and then the STAT is phosphorylated, dimerized, and moves into the nucleus where it activates the transcription. So the regulation of the cell cycle involves a number of different factors, and some of the major ones are shown here. The cyclins, the cyclin-dependent kinases, which form this complex. The retinoblastoma protein, like I mentioned before, was originally found in retinoblastomas, but is present in many, if not most or all, tissues. And P53, again, a phosphorylated protein of about 53,000 molecular weight, are both recessive tumor inhibitors. So if you knock those out, they knock out the prevention of moving into the cell cycle. Also, there's an inhibitor of the cyclic-dependent kinase and some family of transcription factors that are involved. So in the next figure, we see some of these coming into play here in the cell cycle. The cyclins shown in green or blue and the cyclin-dependent kinase in red. And so we see that these different cyclins come on at different times in the cell cycle. Again, they combine with the kinase generally to inactivate them. But let's go to the next figure, and we'll see how these cyclins work and coordinate with each other. And so now we've linearized the cell cycle, and we see 4 cyclins coming up at different phases of the cycle, where the cyclin E, the green one, peaks at the transition from G1 to S, whereas cyclin A is in the G2 phase and cyclin B during mitosis. So you see these cyclins, different molecules come up during different phases of the cycle and promote the progression of the cell through the cell cycle. Okay, so the main restriction point is in late G1, in other words, before DNA synthesis, and this is where you really want to arrest the cells before they go on to start replicating DNA. So growth factors, here abbreviated GF, usually exert their effects between the onset of G1 and the restriction point. So telling the cell to let's go through the cell cycle and divide. So once past this restriction point, the rest of the phases up to mitosis are pretty much committed, even though there is some regulation there. And these recessive genes, the tumor inhibitors, like retinoblastoma, are sometimes called the gatekeepers. So if they're present, they restrict the progression of the cell and through the cell cycle. So again, if we look at the cell cycle, we see the major checkpoint down here at the bottom, G1. Before the cell really starts the process of proliferating, and then again up at the end of G2, There's another checkpoint which holds up mitosis until it's checked to make sure that everything is good to go and ready to divide into two daughter cells. Okay, so this is just a little more detail about the different cyclins coming in and out, as well as the retinoblastoma gene. So just gives you some more details if you're interested. But again, I think just understanding the big picture that these this cycle is highly regulated, and as you might imagine, the major regulation is right before S phase starts, so really before the whole process starts. And then there is some regulation after that, but mainly to hold things up until the cell is ready to divide more than to tell the cell not to divide. So once it's gone through S phase and into G2, it's pretty well committed to dividing unless something catastrophic happens. And so, again, the same diagram, focusing on towards the end of the cycle, where again the p53 is monitoring for damage to make sure everything is good to go. The retinoblastoma gene has been inactivated, and so if things look good, you can send it on through G2 and into M phase. So if we have a growth factor like platelet-derived growth factor, attaches to the cell receptor, initiates these pathways, one through the RAS-RAF, initiating MAP kinase, and another through a lipid signaling pathway, both resulting in transcription of all the factors that are needed to produce for the cell to divide. And again, all these nuclear factors like Jun, FOS, MYC, all are potentially oncogenic if they are mutated in the right way. These genes were first identified because they were somehow picked up by a virus and incorporated into the virus. And then when the virus infected a cell and brought the gene in, it transformed the cell into a cancer cell. Now again, if the cell determines that it cannot go through the cell cycle due to some type of damage, it will change pathways and go through the apoptotic pathway, and the cell will be destroyed, histolized, and probably turned over. The molecules turned over for recycling or gotten rid of. Okay, just a quick look at apoptosis. This death is certainly a part of life. Cells are dying all the time. Most often they can be replaced during development. Some cells are actually fated for death, and that's where this process was first identified during development, where it was seen particularly in very simple model organisms, that some cells went down a pathway that basically led to destruction, so a terminal pathway. Too much of this an inappropriate cell death can lead to some problems, but the subversion of cell death can also lead to cancer or autoimmune diseases. So it's not to be mistaken for necrosis, or the destruction of a cell. This is again a highly programmed process, and these cells will be flagged for phagocytosis. So again, necrosis is when the cell is destroyed by some means, but apoptosis is the programmed cell death. Again, it happens as part of development, and it also happens if the cell decides that it is no longer viable and can't divide. Now, like all cellular processes, apoptosis is regulated, and again in two ways. One, to direct the cell through the apoptotic pathway by these caspases, which are proteases, and then there's BCL2, which is an anti-apoptotic regulator. So we see here in this diagram sort of a competition between apoptosis and growth factor mediated cell rescue. So if there is a problem with the cell, of course it's going to try to rescue itself and induce, if that can be done, it'll induce BCL2 to maintain itself. But if it can't, the caspases are induced and start the apoptotic pathway. Now I want to now shift into looking at cancer a little bit, and we've alluded to the idea constantly when we talk about cell cycle regulation, because it's so important in cancer, and the disruption of it is so important in cancer. But when we think about cancer, you have to realize that every tumor originated from a single mutant cell. So one cell that is mutated to now become carcinogenic or cancerogenic, and grow up into a tumor. Now, when we talked about DNA replication, we remember that there's very few errors during the replication process. Pretty amazing fidelity, about one error in 100 million bases placed. But that's still significant, and because for every human cell that's replicated, there are several errors. Now, many of these could be corrected by the repair mechanisms, but still, every time a cell replicates, there are some differences. Fortunately, most of those differences are going to end up in non-coding areas, since the coding regions only account for a very small fraction of the total genome. And even if they do occur in a coding region, they may not have a large effect on that. Also, it takes several mutations to generate a cancerous cell, anywhere from 3 to 7 mutations, depending on the tissue and what the mutations are. Now, we classify tumors into 3 types: the carcinomas, which are derived from epithelial tissue, the sarcomas, derived from muscle or connective tissue, and leukemias derived from blood cells. In humans, the vast majority of cancers or carcinomas are derived from epithelial cells. Now, remember, there are 2 types of genes that are involved. The oncogenes, or proto-oncogenes, which which are dominant, so only one proto-oncogene needs to be converted into an oncogene to be cancerogenic. And then the tumor suppressor genes, which are recessive, so you need to knock out both of the suppressor genes in order to be tumorigenic, because having a single good copy of the gene is enough to keep the cell under control. Now, originally, oncogenes were discovered in viruses, and it looked like viruses carried these genes into a cell, could infect the cell, and produce a tumor because they were because they had an oncogene associated with it. And this is an example here of the Rous sarcoma virus, which has the Sarc gene incorporated into its genome. And it turned out there are analogs in the host, and these are the proto-oncogenes. So what appears to have happened is the virus infected a cell, and when it left that cell, it carried along with it a proto-oncogene, which then could mutate to become an oncogene. The virus then infects another cell, bringing in now the oncogene into that cell and causing a tumor. Now, viruses can cause tumors in a couple of different ways. The one I just discussed, where the virus can acquire a proto-oncogene and then insert it into a new cell as an oncogene. We can also have a virus infect a cell, integrate itself into the host genome next to a proto-oncogene, and turn that gene on either constitutively or at the wrong time, or at a higher level of expression than normal, effectively producing an oncogene. Retroviruses can also do this by inserting themselves into the genome. Now, in humans, about 85% of tumors arise from point mutations or rearrangements in oncogenes rather than through any kind of viral mechanism. In other animals, that may not be the case. Now, I want to revisit this idea of tumor suppressor genes. Again, these are recessive genes. The retinoblastoma is an example. It's fairly rare, but um, there is a hereditary form which is more frequent, and basically what that means is that the person has inherited one bad copy of the retinoblastoma gene. So, if you have one good one, that's fine, but if you start out with one bad one, your chances of acquiring a second bad one are much greater than actually having a mutation in both copies of the gene. Now, the most common uh, genetic lesion found in human cancer is in p53. Uh, and again, just like the retinoblastoma gene, individuals with only one functional p53 are predisposed to a large number of tumors, depending on where a second mutation would occur. Now, I want to turn our attention to oral cancers. Worldwide, there are about 5% of malignant lesions. The 5-year survival rate is pretty low, about 50%. And most of these are carcinomas, almost all of them. And most are squamous cell carcinomas, which tend to spread pretty rapidly. And this ranks 5th in the global cancer burden, so there's a lot of oral cancers. Treatment could require surgery, radiation therapy, chemotherapy, or more likely a combination. Some of the symptoms associated with oral cancers are red or white patches, a sore in the mouth that doesn't heal, bleeding, loose teeth, which could mean other things as well, painful swallowing, a lump in the neck, or even an earache. So depending on the location and type of tumor, you may get different types of symptoms. This is then an example of early stage squamous cell carcinoma on the tongue. So you can see the lesion there on the side of the tongue. Now, there are a number of risk factors for oral cancers, and of course, at the top of the list is smoking or tobacco use, which is associated with about 3/4 of all cases. The chewing of the betel leaf in some Asian countries really also is a very high risk factor. But there are other factors as well: heavy alcohol use, the virus. HPV or human papilloma virus, chronic irritation, immunosuppressants, and poor dental or oral hygiene. And I should point out generally that actually cell growth it promotes tumors, again because of the mutations that occur during the replication process. So irritations or even surgery can be a risk factor for cancer as well. Now, there are a number of known carcinogens in tobacco, over 90. Some of them shown here. Some of these are metabolites of nicotine, and so. But there are others as well, other aldehydes, which are alkylating agents on DNA. Some aldehydes are actually used as cancer treatment to destroy the DNA. Also aromatic amines or nitrosamines. This is a list of some of the carcinogens or suspected carcinogens, just to give you again an idea of the numbers and variation in these carcinogens that are found in tobacco. Now, what about mouthwash? Alcohol is a principal solvent for other ingredients, and it also acts as a preservative and antiseptic. So it's 10 to 12% in most products, and there is evidence that daily or twice daily use of these mouthwashes may predispose one to oral cancer, and again because of the alcohol. The other risk factor that was on the rise recently, but now maybe going down because most young people are being vaccinated against human papilloma virus. But these, again, are some examples of lesions that are due to the virus. Again, it started to rise in the last 10 years or more. And it has become more common than cancer due to smoking. Some of these strains cause cervical cancer in women as well. The good news is that the survival rates for oral cancer patients with HPV are better than other forms of oral cancer. And just to show you the structure of the virus, you'll probably learn more about this in micro next semester, but this is a typical papilloma virus with some genes expressed. The E genes stand for early. These are expressed early in the infection and code for genes that kind of take over the cell. And then the late genes, L1 and L2, are generally genes that code for the proteins that package the virus so that it can be sent out to infect other cells. So that's all that I want to talk about with the cell cycle and cancer, and we'll move on to next time to talk a little bit about developmental biology.